Good morning and good afternoon. Thank you for joining the first session of our 2020 virtual user meeting. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's presentation. By default, all audience members are muted until the end of the presentation, at which time we would open the floor to some questions. It is my pleasure to introduce Dr. Angelika Lampert from the University of Aachen for the start of the presentation.
Okay, so I'm going to take over. Thank you very much. Thank you for inviting me. It's really a pleasure to be part of the user meeting. And I do look forward to meeting you again, hopefully next year in person, as the user meetings normally are really nice and it's very nice to interact with everybody. Yeah, but this year I'm especially proud because I will soon be able to have the SyncroPatch machine here in Aachen as well. And this is what I would like to talk about today as well. And the title of my talk will be the Sodium Channel Network Aachen meets the SyncroPatch. So this, of course, pops up several questions. The first, it's three of them, I guess. And the first one would be, why sodium channels? Then in the second part of my talk, I would like to introduce the Sodium Channel Network Aachen. And the last question then is, of course, so why now the SyncroPatch? And I hope that in the end I could convince you that it's really a very nice combination to have the Sodium Channel Network Aachen using the SyncroPatch, and we do look forward to having the machine here. Yeah, so why sodium channels? So this person here is walking over glowing coals, and he or she seems not to have pain in that moment while he is walking here. Actually, this is a person who is joining the vegetarian festival in Thailand, and he's taking good care not to sink into the coals here, because if he would sink into the coals, he would feel pain. But there are people on this planet who wouldn't care, who do not have the ability to feel pain, and these people are suffering from a pain syndrome, which is called chronic insensitivity to pain. And it was found out that these people may have a complete knockout of the sodium channel subtype NaV1.7. Now, you may think back, yes, it's a long time ago, our physiology lessons, were these sodium channels good for? And actually, it's really a long time ago. So Hodgkin and Huxley in 1952 published a series of seminal papers on the recordings of the squid axon. So here you see the squid, and in the back they've got the axon where they can easily insert an electrode into. And then they recorded the potential in this axon, and they made recordings of over the cell membrane. And there they saw this rapid change, which they called action potential. And already at that time, they predicted that this quick upstroke here of the action potential was mediated by very quick opening and closing of voltage-gated sodium channels. By now, we know that there are 9 different subtypes of voltage-gated sodium channels in the human genome. And actually, Stummschaltung aufheben. Yes, I think now I'm back on air. So there are 9 different subtypes in the human body, and we found out that especially NaV1.7 seems to be super important for human pain perception. And already in my introductory slide, it was shown that if we have knockouts of NaV1.7, so humans who do not have functional 1.7, that this is leading to chronic insensitivity to pain. But there are also mutations in this channel that lead to gain of function of this channel. And then we've got the inherited pain syndromes, which can be, I'm just mentioning 3 of them, the inherited pain syndrome erythromelalgia, IEM, or paroxysmal extreme pain disorder, PPD, or the small fiber neuropathy, SFN. I now would like to focus in the next couple of minutes on the PEPD, and I would like to show you what you can do with electrophysiology, just normal manual electrophysiology, in order to find out what's wrong in these mutations that lead to PEPD. So what we do is manual patch clamp of HEK cells that overexpress ion channels that carry a mutation. In this case, it's the A1632E mutation that is linked to symptoms of PPD. And when you do so, you can first, of course, you look at the wild type channel. And there you see that when you are changing the voltage, you get this nice inward current and almost no persistent current at the end of a 40 millisecond pulse. But when you're doing the same with one of the PPD mutations, what is quite characteristic for this disease is that you do get a pronounced persistent current. This is quantified down here, so you get quite a lot more persistent current in the PPD linked mutation. So there seems to be impaired inactivation, which then results in increased persistent current in these channels. So how is this happening? How can an ion channel actually have inactivation, and how can this be impaired by one of these mutations? So one of the really nice advances in our field was that it was found out how the structure of the sodium channels is looking like, and it's actually looking like this. So here we've got a 3D print of one of these voltage-gated sodium channels. So most likely to many of you, it just looks like a blob. So in order to make it a little bit more understandable, I brought a picture of it. So what you see here is a picture of it from the side. I think it's this orientation. Yeah. And what you see here are these little things sticking out. And these are the voltage sensors here marked with the arrows. So we've got 4 of these voltage sensors. I think this is the best to see. Yeah. Looks a bit like a helicopter. And these voltage sensors are linked to the pore region, which is up here. And in my little model here, I put a cord through the pore region. So this is where the sodium ions would permeate the sodium channel. Okay, so it sits in the cell membrane like this. And then upon a depolarization, the voltage sensors move in the electrical field. The pore opens, and then the sodium ions would permeate through the channel into the cell. Now we can make a computer model of the 3D structure. And if you now tilt the channel like this and look at it from the intracellular side, it looks in the computer model just like this. So this is a computer model which we have done in the lab by Janis Körner, and it is based on this publication of the 3D structure. And again, you see here the 4 voltage sensors. And in the middle, we've got the pore region, and when the voltage sensors move and the channel opens, then the ions permeate here through the open pore. Yes, so normally what we thought is when the sodium channel opens, it very quickly inactivates, and we were thinking that this is happening via a mechanism which is called the ball and chain mechanism. So somewhere on this ion channel underneath, there would be a linker, which is maybe not represented in the structure here, which then would swing into the open pore and block it. So that's the pore, and this is the so-called inactivation particles. So these red amino acids, which you see here, they are thought to be the ball, which then would block the ion channel. So here it's a bit more schematic. Here we've got the pore, the sodium ion flows through the pore. Then we've got the so-called IFM motif, isoleucine, phenylalanine, methionine, which are these 3 amino acids. And once the channel opens, within milliseconds, it would inactivate and the sodium ions would stop flowing. Now, with this new structure and several structures following this 1.7 structure, or some of them even published before that, we started to think that there could be an alternative inactivation mechanism. And that is the so-called allosteric mechanism, which is now proposed instead of the hinged lid mechanism where the little ball is swinging into the pore. So how come? Here you see the amino acid A1632, and this amino acid seems to be a part of a binding pocket of this inactivation particle, which is here depicted in red. And the idea is now that the inactivation particle would actually bind from the side of the channel. So here you can see it. This would be the inactivation particle. And as you can see, it's not sticking out of the channel, but it's sitting at the side of the permeation pathway. And it seems to push the permeation pathway from the side and push it close it. Now, the mutation that we were looking at, the PPT mutation, seems to interfere with fast inactivation. And interestingly, this amino acid A1632 is exactly the amino acid which was mutated in in the patient that we were looking at. And now Jannis Körner, here you see him with his doctoral hat together with Annika Rühlmann, who did all the electrophysiology experiments of that. They looked at this area a little bit more closely. So this is a zoom on of the permeation area. Here, down here, you find the inactivation particle, and this is the A1632, which is mutated in the PEPD patient to an E, so an introduction of a charge. So here we've got the A1632. This is the permeation pathway, and down here we've got the inactivation particle. And you can see that the inactivation particle can nicely enter this binding pocket and then shift this S6 segment of domain 4 towards the inactivation, the permeation pathway, and stop the ions from move, like inactivating the channel. Now let's look at the situation when A1632 is substituted with E. And already here you can see that the E is much larger compared to the A, and we almost have a steric clash here between the inactivation particle and the mutated residue on position 1632. And now it's much harder for the inactivation particle to bind and to prevent permeation of the ions by shifting the domain 4S6 segment. So it seems that the PBD mutant A1632E hampers fast inactivation by steric repulsion. But in addition to introducing a larger size of amino acid, it also introduced a charge. And this could also add to this quite prominent persistent current that we get here. Now let's look at the charge, and you can do this by looking at the hydrophobicity of the binding pocket. And this is the wild type. Here we've got the A1632, and you see it's quite hydrophobic, which is nice for the IFM motif to bind. But when we introduce the inactivation particle, but when we introduce the charged glutamate here, glutamic acid here, you can see that now the hydrophobicity also changed in this binding pocket, and now it's much harder for the IFM motif to actually bind in the pocket. So we've got two reasons why the IFM motif may not bind as nicely in the mutated channel. The first reason could be that we've got a larger amino acid and therefore steric repulsion. And the second reason is that we have a changed hydrophobicity, and this then also reduces the binding of the inactivation particle. And these experiments also support the notion that we have a inactivation which works via allosteric mechanism and not via the so long thought to be true ball and chain mechanism. So Annika continued her experiments on this mutation by co-transfecting wild type and the AE mutant into HEK cells. And what you can see here is that when she was looking at the persistent current plotted here, that when we have the co-transfected mutant with wild type, you see that the persistent current is quite small compared to the AE by itself. It's not really sitting in the middle between the two values, but it's smaller. We could also repeat this in Xenopus laevis oocytes. This was done by Ralf Hausmann and Günther Schmalzing in the pharmacology here in Aachen. And both of them show that actually when you have the two constructs present, it somehow seems to be less persistent current than when you've got just the mutated construct present. So is it possible that the two channels are interfering somehow with each other? And now we come to the construct of dimers of sodium channels. So the sodium channel is quite a large chunk of ion channel. And for a biophysicist, it's quite hard to imagine that this large chunk of ion channels should form somehow dimers with a second ion channel being there, sodium channel being there. And so therefore, up to now, everybody was kind of rejecting this idea that sodium channels could be dimerizing, especially because these chunks on the side, they are moving really quickly in the electric field. But then there was the group of Isabelle Duchesne, Plateau et al. They published in last year in Nature Communications a story where they showed that NaV 1.7 seems to gate as a dimer. and it seems to interact with each other by a region in the area, in the intracellular area at the elongation of domain 1s6. And this area here, which is marked in red, seems to be linked to each other of two sodium channels by the protein 1433. This is shown here. This is the protein 1433, and there seems to be not only one dimerization site, but two. So there seem to be two 1433s that bind two sodium channels to each other. Now there's an antagonist of 1433, which is called D4P, and it is able to bind it bind into the 1433. It's these arrows shown here, and you can co-transfect it and then check whether actually your dimerization is still happening or not. And they could show that when they co-transfect D4PE, the effects of dimerization on NAV 1.5 were quite reduced. Now the question is, if NAV 1.5 is dimerizing, is maybe 1.7 dimerizing too? This we discussed with our colleagues of the pharmacology department, and Günther Schmalzing there was actually running a gel. And what he was doing there, he was using NAV 1.7 with a GFP tag, and he was using, several different detergents. And he could nicely show that we get this band at the size of about the size of the monomer. So this is what we expected. But in all of his preparations and in all the different detergents that he was using, he could actually see that there are also bands that correspond to the dimeric size of the sodium channel. So it seems quite likely that we actually also have a dimerization of the voltage-gated sodium channel NAV 1.7. Here, here you see a control of TRPV1, and you see that it's nicely having its 4 subunits coming together. So this for us was interesting, surprising to a certain extent, and now we wanted to see whether these dimers are actually gating in a different way compared to the monomers. So what Annika did was that she was using HEK cells that express A1632E by itself. Now, there is a lot of internal 1433 in almost all of our cells in the body. And so we need to assume that in these HEK cells expressing only NAV 1.7 with this AE mutant, that they are there as a dimer. So this large persistent current that we see there is most likely because we have dimers in there. Here we've got the persistent current quantified. Here are the experiments performed also in Xenopus levis, where we've got this large persistent current. Now she was co-transfecting, or it was co-injected. This was our experiments by Ralf Hausmann, and we co-injected then D4PE. And D4PE, that's the antagonist of the dimerization. So the sodium channels should be there as monomers now. And you can see that immediately the persistent current is declining, not only at 1 voltage, but over a large range of voltages. And you can see that this is not only in HEK cells, but we could reproduce it in Xenopus levis oocytes, where you also can much better control the amount of cDNA you're injecting. So the persistent current of the A1632E mutant, which is linked to pain, is enhanced by dimerization. So we could show that not only that dimerization occurs in 1.7, but it seems also to be relevant to certain diseases of mutants that are affecting, in this case, fast inactivation. So one of the questions of my talk was, why sodium channels? And I think I could answer, I hope I could answer that. They are super important because they are linked to human genetic pain syndromes, which could help study human pain. But not only that, I think they are also very interesting when it comes to their biophysical gating and structure-function relation. And with the experiments I just showed you, I hope I could convince you that sodium channels fast inactivation occurs most likely via an allosteric mechanism and not via the hinged lid or ball and chain mechanism. NAV 1.7 seems to form dimers, and you can interfere with this dimerization, which then reduces the PPD mutation-induced persistent current. So you see, sodium channels are really quite an interesting ion channel to look at, and I'm pretty sure that Fernanda Laesa will tell you a little bit more about the plethora of implications of sodium channel mutations in physiology and pathophysiology. Now we come to the next point. What is the sodium channel network Aachen? So the Sodium Channel Network Aachen has this nice sign, and it's a consortium which is funded by the local IZKF, Interdisciplinary Center of Clinical Research here in Aachen. And it is a combination of several highly interested researchers that have the following aim. They want to study sodium channel related pain disorders, and we would like to cover the whole range from molecular mechanisms towards personalized treatment. And in order to do so, we have grouped these different projects together. And the idea is that we would start out with the patient and its mutation, its clinical pain characteristics, that we check for the genetics, check for the effects of the mutation on the channel itself, structurally, but also functionally, and then get ideas that could maybe help the patient in the end, and also potentially treat in the future the symptoms of the patient. So in order to do so, the first thing that you need is clinicians that are willing to work together with basic scientists and basic scientists that like to work with clinicians. And I think we are really lucky here in Aachen that we have a very large and highly motivated group of clinical scientists and clinicians who are actually interested in working with pain patients and characterizing them. It not only comprises the neurology department and the pediatric neurology department, but also the fMRI studies and with the palliative care and the IZKF Nachwuchsgruppe of Barbara Nama, people who are actually doing microneurography or QST experiments on patients. And with these really nicely characterized patients, we now can draw blood, and especially when we have a family history, we do the genetic analysis with the human genetics department, and the variants which are identified there are then submitted to testing. And we've got a whole group of electrophysiologists who are looking forward to do these testings and are actually already doing these testings of the mutations identified in the patients. We are not only looking at the mutations, but also at the variants in some cases. So Ralf Hausmann, here he is. He is doing single channel recordings, Günther Schmalzing, the biochemically sodium channel analysis. But what we also do is we are creating iPS cells, induced pluripotent stem cells of the patients, and derive nociceptors in order to do patch clamp analysis. Now we get an idea of the function. Ideally, we would also like to get an idea about the 3D structure and how this may change drug binding. And therefore, we've got Julia Rossetti and Jan-Philipp Martens who are then doing the modeling of the mutations in the 3D structure. And we also do some in silico drug development. So this is really nicely spanning the whole picture from the patient all the way to the mutant in the 3D structure over the function and genetics behind it. And the drug development in our hands until hopefully only next month happened only in silico. And the question which is raising here now is, where is the high throughput electrophysiology drug screening? Because now if we have this large cohort of 200 and plus pain patients, very well characterized, genetically analyzed, we now have really the chance to find mutations or variants that may react differently to different drugs, or where the iPS-derived sensory neurons may react differently. And it would be perfect to have a high throughput drug screening available in order to check for that. And this directly leads to the third question, why now the SyncroPatch? And I think it doesn't need much explaining that this is the tool which is desperately needed in the moment, and we are extremely happy. Together with Ralf Hausmann, we wrote this application and that we've managed to win the 9 months of SyncroPatch here in Aachen. So what do we plan to do with the SyncroPatch? So first of all, of course, we need to develop the assays, and we are looking forward to the help of maybe even Martin Hampel, who's maybe used to Aachen more than anyone else at Nanion. And once we have the electrophysiology assays developed, then we can check for the biophysical changes of the sodium channels much easier in the SyncroPatch. So we do the characterization of the pain mutants, but not only at room temperature, but also at different temperatures. And that is especially important because quite often the pain patients have the problem that when the temperature outside is raising, their pain attacks are triggered. But we don't really understand fully this interrelation between temperature and the biophysical changes yet. So it's perfect that with this machine, we now can look at this in a bit more detail. And then, of course, what we will make mutants that will help us to understand the 3D structure of the sodium channel, which we can then do molecular dynamic simulations to understand in more detail the inactivation gating, or maybe even activation and deactivation processes. And now that we have this biophysical characterization, now we can go one step further and check for drug sensitivity of the different variants and mutations of the sodium channels. We do this in HEK cells or ND7 cells when it comes to sodium channels that express well in ND7 cells. And what we would like to do is we would like to take the spectrum collection of drugs, which we already ordered and it should soon be delivered, in which we have all the FDA-approved drugs, and we would like to screen them on the mutations that we found to be electrophysiologically relevant in the SyncroPatch. Once we have hits in the screen, then we would like to validate these hits in pig and mouse DRGs. And there are already protocols where how we can use these pig and mouse DRGs at least in the SyncroPatch. And we would like to use pig because pigs are much closer to humans, especially when it comes to the C fibers that are most spontaneously active in neuropathic pain. The pigs seem to have them, whereas the mice seem to lack them, at least in the skin, where it seems to be most important for the neuropathic pain patients. So we would like to widen the protocol to pig DRGs, but of course pig is not human, and ideally we would like to try also to get the iPS-derived sensory neurons of selected patients and do the analysis of the identified compounds either by hand with manual patch clamp or ideally, but this is still some work to go, also on the SyncroPatch. So we are very much looking forward to receiving the machine. I would like to thank my lab in Aachen, my cooperation partners, especially Ralf Hausmann, with whom together I wrote this application for the SyncroPatch. Here is my funding, our funding here. And I would like to thank you for your attention. And of course, we are open for questions. Thank you very much.
Thank you, Angelika, for that nice presentation. I kindly will now moderate any questions from the audience. I would just ask that perhaps you would type into the chat box if you wanted to ask a question, and then we can unmute you. I think there's a question from Ron, perhaps.
Yes, please. Ron Knox in the United States. Can you hear me,
Yes,
Angelika? Oh, great.
Yes.
I have a couple small questions. I was wondering if you had any evidence that the NAV 1.7 dimers contribute to the persistent NAV 1.7 currents that you record from animal dorsal root ganglion neurons.
Yeah.
And then further, and the other question would be, what would be the implications if this were true for the pharmacological management of pain, for example?
Mm-hmm. So up to now, we only did these experiments in HEK cells with D4-PEIN. We did not do D4-PEIN transfection into DRG neurons yet. So I cannot answer your first question. These are experiments to come. The second question about the therapeutic potential of dimerization is it's not very good because 1433 is simply expressed in almost all of our cells. It seems to play a really important role in immunology. And of course, you don't want to mess with immune system about that. So it seems to be rather a biophysically thing, and maybe we can find drugs that would interfere with dimerization in the end. But it's not that 1433 is a good drug target, not at all. One more thing that I would like to add to that is that we tested NAV 1.7 wild type with and without D4PIN. And if you have D4PIN present, so if you have monomers, the inactivation seems to be shifted. So dimerization seems to have an effect as well on the wild type NAV 1.7. Yeah.
Okay, great. Well, thank you very much.
Most welcome.
And I think we received an additional question from Dr. Sandeep Padar. He asked if there was any effects of diet on this channel.
Actually, the honest question is, I do not know. Most likely, diet can also affect the trafficking and the modulation of the channel, glycosylation of the ion channel as well. But I don't know about it.
I'm just opening up the floor again to any additional questions that may have. And if there's nothing that you can think of immediately, then we welcome you to get in contact with us, write us an email, and we're happy to pass it on to Angelika. And for those maybe on different time zones that weren't able to join live, we're going to transcribe this and make it available on demand. So in about 24 hours, the presentation will be available as well. There was one more question from Dr. Pierre Debs. Regarding iPS-derived DRG cells, what cell type is being reprogrammed?
We are reprogramming both. We used to reprogram fibroblasts because they seem to be more stable, but by now we are doing it from PBMCs, so blood cells, white blood cells, and that works equally nice. So that both of it works well. Yeah, we may think that the fibroblast-derived iPS seem to be a little bit nicer for differentiation into sensory neurons.
And then I think we might have another question from Ali.
Okay.
I'm not sure, maybe she's unmuted herself or... Okay, so I will move on to another question from Diego Fernandez. Is SyncroPatch dealing properly with eventual series resistance problems that might arise when recording fast NAV 1.7 currents?
So actually, I do hope so. I don't have the machine yet. But I mean, what I learned from demos and last user meetings, it seems to be quite good when it comes to series resistance. And to me, so what is really nice is this high gigaohm that you get. And then when you have large pipette holes, then I hope that the series resistance will be okay. Maybe somebody from Nanion can comment on that better than me.
Okay, and then an additional question, do you check for re-expression for NAV subunits before and after reprogramming?
The re-expression, yeah, so we do check the sodium channel expression, of course. We do patch clamp, so we functionally see whether the sodium channels are there. And of course, we also apply TTX in order to see whether TTXR currents are there. And right, and then we also check for peripherin expression in every differentiation that we do, because we realize that there's large clone-to-clone variability, and also differentiation to differentiation varies. So in parallel to patching, each differentiation has a quality control step, which includes staining for peripherin to see that we really have peripheral sensory neurons.
And then a question from Ivan Katerin from UCL. He was wondering if dimers form via interactions from transmembrane domain regions or cytoplasmic domains, and if you ever separated the NAV homologous repeat domains of the channel to map.
Yeah, so I really like your questions. Thank you. Thank you also for listening in, Ivan. These are the questions we are following up. So the paper of Plateau et al., they suggested that the interaction site would be somewhere intracellularly, so somewhere here, and not via transmembrane interaction, which also makes much more sense, because these are the voltage sensors that are quickly moving, and it's quite hard to imagine that they would interfere with another ion channel. Nevertheless, it's not impossible, you never know. So what we are doing in the moment within the sodium channel network, we make large-scale computer simulations on that. And we are also trying to find some ideas from these simulations to do screening mutants to interfere with this interaction. And especially with this 1632E mutant, we now have a tool with which we can see whether dimerization still happens or not, functionally see. And especially now with the SyncroPatch, we also have a tool to make it quicker, give our electrophysiology a boost and make it everything much quicker. I hope so. Yeah, but definitely this, I think it's super interesting to find out where the direct interaction is. Thank you very much.
So thanks again, Angelika, from us here at Nanion for presenting this. We're all very much looking forward to your group getting underway here. Again, I'm going to transcribe all of this, and it's going to be available latest 24 hours from now, so that can be shared across all of your networks. And we thank all of you for joining us. I know it's early on the East Coast, and maybe those in the Pacific also joined in, a couple names there. So thanks so much, Angelika, and our next session will begin in a couple of hours for Fernanda's talk.
Thank you very much. Thank you for inviting us.
Have a good day. Take care. Bye-bye.